Hi everyone, my name is Amalia and I'm a first-year PhD student. My project is concerned with the biomechanical control of ovarian follicle development, and my supervisors are Dr. Ian Dunlop, Professor Kate Hardy, and Professor Stephen Franks. My project aims to address a fundamental issue of cell culture, which is that cells in vitro grow in a two-dimensional environment, which does not provide them with the mechanical support and stimuli that they have in vivo, which leads then to results that can differ a lot from what actually happens in the body. So why? Why is this question interesting or significant? One area of significance is that of ovarian follicle culture. Large numbers of women suffer with polycystic ovary syndrome or premature ovarian insufficiency, both of which lead to fertility issues as well as disturbing other functions of the body. Currently, they are being targeted with treatments that mostly use drugs or hormones, but biomechanics are increasingly considered in the development of new treatments. So, the hypothesis behind my project is that a composite hydrogel culture environment that mimics the microenvironment of the ovary could present an accurate in vitro model to help us understand better the ovarian pathologies and later on to discover treatments, which means that my goal is to design a composite hydrogel that has mechanical properties similar to that of the ovary, to fabricate it, and finally to test it and see its efficacy in cell culture. In previous research done in our lab, the mechanical environment of the ovary has been characterized, and it shows considerable variations in stiffness from the center to the most outer regions, as it can be seen from the graph. These findings also suggest that stiffer regions in the areas with big follicles and more compliant regions where the small follicles and more extracellular matrix lie. So how can we use this knowledge to create a material that mimics these properties? In my project, I use hydrogels because they have quite a big range of different stiffnesses and the ability to provide cells with nutrients. The composition of the hydrogel is also quite important since it needs to mirror the heterogeneity of the ovarian environment, so maybe a composite material could achieve that.